The next speaker is Dr. Antonia Pritchard, and she's from the Queensland Institute of Medical Research, and she's looking at current research in oncogenetics. Well, I can't pronounce it as a Dutch person, sorry about that. Dr. Pritchard is a postdoctoral staff member of the Queensland Institute of Medical Research Oncogenetics Laboratory. I'll work on that one over the lunchtime. The role of the laboratory is to identify novel cancer genes and study the way in which defects in these genes are associated with cancer predispositions or development. Dr. Pritchard and the oncogenetics team has the interest in melanoma research, focusing on melanoma susceptibility genes. And we would welcome Dr. Pritchard, who will today be providing us with an overview of the current melanoma research being undertaken at QMR.
Good morning, everyone. Can we all hear me okay? Okay. So I would like to thank Melanoma Patients Australia for giving me the opportunity to come and talk to you today about the research that we are undertaking at QIMR. I've got a little bit of history about QIMR here. So it was established in 1947, so just after the war, when there was an outbreak of Q fever in North Queensland. And this was our very first laboratory. It consisted of 7 staff members. And it stayed on this same site actually for about 30 years. We've changed slightly since then, and this is our new buildings. So this far building over here was opened in 1990 and was very state-of-the-art at the time. This new building here was opened in 2001, and this is where we reside in the oncogenomics group. And then this middle building here has just opened in 2 months ago. We've moved in. I think we have the official opening in 2 weeks' time, which is absolutely state-of-the-art, beautiful, beautiful building. This building, the poor little Bancroft Centre, is now being sort of refurbished and brought back up to date with the other 2 buildings. The site at Hairston has the Royal Children's Hospital next door, and then at the other side here we've got the Royal Brisbane Hospital and Royal Brisbane Women's Hospital. So we're ideally situated between many hospitals, which is a good place for a research centre to be. So at QIMR, we currently have about 700 members of staff. So we have scientists and we have scientific support staff. So scientific support staff are vitally important to the work that we do. They help run facilities and that kind of thing. And we have a big admin team as well who are involved with lots of things, some of which I'll tell you about in a minute. And we also, on top of that, have 110 students working at QIMR. So they're both honours students at the end of their degree and then PhD students who are starting out in their research careers. So I'm sure that we've seen a lot of these kind of graphs here this morning. I just want to show you this to reiterate again how important melanoma and skin cancer research is within Australia and New Zealand. So as you can see, the incidence in Australia and New Zealand is significantly higher than the rest of the world. And I think that the amount of labs and the amount of research devoted to skin cancer and melanoma within QIMR represents this real need within Australia to research this disease. So within QIMR, we have got 3 main divisions. So there's a cancer division, a mental health division, and an infectious diseases division. And then within the cancer division, we have this Skin Cancer Research Centre, which consists of 10 main areas of research, which is carried out essentially by 13 different research groups. So you'll probably recognise some of the names on here. This is Adele Green, who has just won the Queenslander of the Year this year and is therefore nominated for Australian of the Year, which is a phenomenal achievement, and I think really is in recognition of the work that she has done in this area, and it's an amazing achievement. I don't know if many of you will know about Jim Aylward. He has quite an interesting story. He lost his job with CSIRO, it was about 10 years ago, and approached Peter Parsons in the drug discovery lab saying that he had this traditional remedy for sunspots and basal cell carcinomas, which is from this sort of sap of this plant, which is a common weed in Australia called radium weed, I think it's called. And so he approached Peter Parsons and said, look, I really want to work out if there's actually something in this traditional remedy. What components of this plant has an effect? So Peter Parsons took him in, and they sort of went through the process of working out the active components within the sap of this plant. And 2 years ago, have produced a gel that you can put onto sunspots and basal cell carcinomas, and they actually shrink them and reduce them. And so this year, in recognition of Jim Aylward's just phenomenal drive, he won the Translational Research Excellence Award this year, and he's a really lovely man, so it was definitely very well deserved. And then that brings us on to us. So this is Nick Hayward, who is my boss, and he runs the oncogenomics research group at QIMR. If you're interested, I think he's going to be on the ABC News this weekend. They did an interview on Friday, so you can see him talk in action. So I thought I'd put this picture up just to show you what the inside of a laboratory actually looks like. I think we've all seen far too many CSI programs, and sadly our laboratory doesn't look much like CSI. Slightly more cluttered. We have an awful lot of stuff on shelves, and we don't have any of those fancy computer screens that you can touch and move stuff around in. I'd put a picture of Ken in here. Ken was a PhD student who's just graduated, and this is him hard at work. Actually in daylight hours for Ken, which is quite unusual. He's very much a night bird. And then finally, there's a picture just of our group here with Nick, and I will go through and talk about the research undertaken by each of the members of our group, but I would particularly like to highlight right now the 2 research nurses who work with us. So this is Jane and this is Judy, and they play an integral role in our group, talking to patients and recruiting them into various studies that we undertake, and they are both incredibly hardworking, very lovely ladies. I will give their contact details later. So, as we know, there are a number of risk factors associated with the development of melanoma. It is a multifactorial disease, and in combination, these risk factors can lead to melanoma developing. So, in our group, we're obviously interested in the genetic side of these things, but it is important to keep the genetic side of things in context with the environmental components that affect the genetic components becoming important within the disease. Now, this is an incredibly complicated looking slide, but I'm not going to tell you about how these machines work. But I put this in here to remind me to tell you that genetic variation is completely normal. So our eye color, whether our earlobes attach to our heads, how our immune system works, all of those things are caused by genetic variations, and they are completely normal. And we have thousands and thousands of them in our genome. And recent technological advances in the last 5 years have made an amazing difference to how we understand genetics and how we can actually research genetic variations within people. And it's machines like this which can very, very quickly, like within half a day, can type in a person hundreds and thousands of these variations. And this is literally, the last 5 years has been a phenomenal time to be part of genetic research, because we can now do these experiments that we could only dream of when I started my PhD. I used to do these individually on little gels, and now we can just run these chips and they do hundreds of thousands of variations. So that's a very important point, which I will come back to. So these are known variants within the genome and can be screened very quickly using these gene chips. even more recently, there is now technology where we can screen the entire genome and sequence the entire genome. So I'm sure that most of you heard of the Human Genome Project several years ago now, where they did the first sequencing of the entire human genome. And at the time, that was an achievement unlike any that we actually thought that we would see, given the technology at the time. And the Human Genome Project actually pushed forward technology to make it easier to do the sequencing. So we started off doing very, very small fragments of the genome at a time, and it drove the need for these changes in technology, and really brought forward the technology such that when the genome was initially screened, I can't remember exactly how many millions of dollars it was, but it was a lot of millions of dollars for one genome that it cost. And now we have these machines actually at QIMR. So we have one of these and one of these, which can do an entire genome for a couple of $1,000s. And it's amazing that we have reached that stage in such a short period of time. So we can quite literally take a person's DNA and sequence every single base pair in their DNA. It's obviously a very labor-intensive procedure, so we mostly use this to discover new variants within the DNA. So we've got the gene chip technologies, which screen for known variants, and we have this sequencing technology, which does new variants. So, as I say, this has been an incredibly exciting era to be in genetic research, and this is just a selection of papers, most of which have Nick's name on them, to have come out in the last 5 years about melanoma genetics and pigment genetics in particular. And the identification of these genes has been driven massively forward by this increase in technology, and I can't reiterate enough how much of a role this technology has played in driving forward our recent discoveries, and it really is an incredibly exciting time to be involved in the genetics field. So I will go through kind of people who work within our group and give you a bit of an idea of the type of research that we do. So there is a very small fraction of people who develop melanoma who have a very, very strong family history. Now, these family histories are generally caused by mutations within genes that are passed on in the families. Now, these mutations are different from kind of normal variations within genes, and they are pathogenic. So there's a small percentage of melanoma cases which have this family history. So mutations in 2 genes have been discovered and categorically account for 50% of this melanoma, which has a familial basis. So this is Lauren, and she is investigating other causes of familial melanoma. So we've got 50% caused by these 2 genes, so there's 50% unknown. And we have very large cohorts of families that don't have mutations in either of these genes that she is currently investigating for new genes. The importance of working out these genes sort of can't be underestimated. So although it is a small number of melanoma cases, actually finding out the genes that are in pathways that cause melanoma to occur lead us to then find genes associated with those pathways in sporadic cases. So these 2 genes, which are mentioned here, they do also play a role in sporadic cases. So that's me. Slightly different hair color back then. So, like I say, sporadic melanoma is the kind of more common cause of melanoma. And some people are more susceptible to developing melanoma when they are exposed to the normal environmental risk factors. So you have some people who sunbake and will, and some people who sunbake and won't develop melanoma. So what I'm essentially doing in the lab is comparing very large cohorts of people who have melanoma compared to those who do not for normal variants in genes. And those variants in genes that are increased in the cohort of melanoma patients could lead to an increase in susceptibility to melanoma. So it's using that gene check technology where we screen thousands of patients for hundreds and thousands of variants, and we see if there is a disproportionate amount of those variants in melanoma patients compared to healthy controls. Like I say at the bottom here, the discovery of these genetic variants can lead to identification of the most at-risk individuals. And in the future, though we are way away yet, we do hope that we can identify people who are more at risk, and they can then be flagged for increased screening and, told to keep out the sun and that kind of thing. So that is kind of the future, we hope, of identifying these sporadic genetic risk factors. So in cancer research, we have the susceptibility actually to develop the disease itself. So you look in the blood for those variations. However, your tumor itself actually develops mutations of its own, and they are vitally important to examine as well, because they drive changes in the tumor itself by these mutations, and the mutations accumulate within the tumor. So the way that we can look at the differences between the tumor genetics and the genetics of yourself is looking at blood and comparing it to the melanoma, and then seeing what the differences are. And that's how, for example, the BRAF mutation was discovered. It is not present in the blood, but it is present in the tumor. So this is Ken. I think he's cut his hair since the picture in the lab. So when he finished his PhD, he became more respectable. So he is involved with looking at particularly melanoma tumor genetics. As you all know, the identification of these tumor-specific mutations has led to the development of new therapies, such as the BRAF inhibitor. So Ken's research project is essentially to identify multiple common melanoma-specific mutations. So BRAF is by far the most common. So when I'm saying common, we're sort of talking 25% of melanoma containing those mutations. But if you can identify multiple amounts of the 25% of people have those mutations, then you can start to combine therapies together. Melanoma can consist of different populations of cells also, so it's important to try and identify different populations of cells which can then be targeted by these different therapies. So the first step, before the clinical trials even sort of get going, so that's a long process, we have to come right the way back into the lab and actually identify the differences between the blood and the tumours. So the next step, after you've actually identified these mutations between the blood and the tumours, is to work out what the function of those changes are. So, for example, the BRAF mutation causes an increase in that gene to be produced. So once Ken has identified these new common mutations, Vanessa and Mike, they take it into the lab. Now, I put this up here because I'm guessing that people don't know exactly what we do in the lab. So patients who have donated their tumours, the tumours can be established into what we call cell lines. So essentially we have grown the tumours in the lab, and we can then manipulate those tumours that we have grown in order to investigate the different mutations and what they do. So that's what all the pink stuff is here. So these are cells, and they grow on the plastic surface of these flasks. And when you look down the microscope, this is what they look like. Not all cells look the same, not all tumours look the same, but this is just an example of you can grow your tumours in the cell, in the lab, and look at them under the microscope and discover what different mutations can do. So knowing these functional effects has two potential outcomes. I say potential because it's what we hope will be an outcome. So the better understanding of the process of melanoma development and also of the development of metastatic disease. And also, again, developing new targets for these therapies. So the BRAF mutations is a really good example of a drug which was identified in the lab, developed, put through clinical trials, and is now available. It's a long pipeline, but it is, something that has now got proof of principle behind it. So I was laughing earlier at Mark's picture of his of his cells with his dendritic cells. This is my version. It's not quite as fancy as his. So this is again a project that I take part in, which has been sponsored by the Ride to Conquer Cancer, who've been a massive supporter at QIMR of cancer research. So essentially what we've got here is a normal cell which produces, so this is normal protein being produced, and so the cell just chugs along happily, it's producing the correct proteins. So in the case of a mutation, what normally happens is that the cell itself detects that a mutation has occurred and corrects it. And that is what happens in normal cases when mutations occur within your cell. However, there are times when mutations escape detection by the cell and escape repair by the cell, and then require secondary defenses to these mutations having occurred. So the consequence of mutations occurring are these abnormal proteins being produced. So the abnormal proteins are signaled by the cells to the immune system to signal, as Mark said, come to me, destroy me, I am abnormal, is essentially what the immune system is hearing from these cells. So my project is aiming to identify mutations within an individual that actually elicit an immune response within a patient's blood. So we've got tumour samples and we've got blood samples, and now this amazing sequencing technology has really come to fruition, and we can identify all of the mutations within a person. We can now start to investigate whether those mutations do cause an immune response. This project is in very early days, but if we can streamline a pipeline where we can identify mutations and then identify the way that it can signal to the immune system, it has amazing potential as a personalised immunotherapy. I say it's very early days, but we're hopeful that it will actually be successful. And so finally, this is Mitch. I think a lot of you met Mitch last night. He came to the cocktail evening last night. He's a PhD student currently, but he has been an RA within the lab for 14 years before he decided to embark on his PhD. So he is highly devoted to melanoma research. He has been in it for a really long time. As somebody said to us last night, couldn't believe that Mitch had been around long enough to be in research for 14 years, but he has. So his research is focused on the identification of biomarkers. So as Mark was saying earlier, it's an important step if we can identify biomarkers that can be easily measured from the blood that ring an alarm that there are tumour cells present somewhere. So Mitch's theory is that there will be biomarkers present within the blood that you can screen for that will announce that it is metastasized or there is a recurrence of disease, etc. So that's Mitch's project, and he has some really promising targets that he is currently investigating in a much bigger cohort of people. So he's got very promising results from a small cohort of people, but he's now expanding his results. So this is my little mini plug for Mitch's research. if you or anybody you know would like to take part in this research, either as a healthy control or as a melanoma patient, he is crying out for people. It's just a blood sample, and he would really appreciate it. So Jane Palmer, who is one of the nurses within our research group, would happily take your call and will answer any questions that you have. It's also on the Melanoma Patient Australia website under the latest news section. It's a big long letter which explains more about Mitch's research, and if you could read it, that would be amazing. So I'd just quickly like to acknowledge Melanoma Patients Australia for inviting me here today, and then also our sources of funding. Medical research funding is hard to come by sometimes, and we are in incredibly lucky position at the moment to have sufficient funding to take our research further. And finally, these are just some contact numbers if you have any questions or would like to know more. Thank you very much.
Are there any questions at the moment about genetic? No? Good. That's a good.
Easy.
Correct.
Kind of off the topic, but does your research cover like telomeres? Do you look into that?
We don't personally do telomere research. I'm trying to think if anybody at QIMR does. I'm not sure that anybody at QIMR does either. There are research groups in the US who are doing a lot, but I don't think anybody here does.
Thank you very much.
Thank you. Thank you.